Marhaba everyone, my name is Sinai. I'm a co-founder and machine learning team lead at Dyno Therapeutics, and I am excited to talk to you about machine-guided discovery, which is an emerging field in machine learning. So the agenda for today is I will talk a little bit about machine learning successes in the past decade and why I think they have been happening. Second, I will talk about the machine-guided design field, which is an emerging field with its own unique challenges. And then I'll talk about how Dyno's problem is of that kind and what problem are we working on. And finally, what is an overview of what our approach is. So in the past decade, machine learning has had many successes. For instance, in modeling, we know now that machine learning algorithms can do a very good job at detecting objects inside images or detecting faces, or we can do good translation between languages or generate human-like text. And recent newsworthy discovery was the ability to computationally fold a protein to its structure based on its sequence alone with some additional information. Other examples are doing really well or better than humans in board games like chess and Go, or video games. So why has this success come about? One reason is that we can do a lot of computations now easily, and some new architectures have also been designed. But the main driver, in my opinion, is the availability of a lot of data, particularly the kind of data that you see on the internet, for instance, images or language. There's a lot of text on the internet. Obviously, the internet is biased, and the type of text and images you find there are biased towards particular ethnicities or particular languages. And a big part of doing machine learning today is to try to overcome those biases. Other cases of success, for instance, for protein folding, is that you had a lot of data, not necessarily a lot of protein structures, but very well curated protein structures that were part of a competition for a very long time. They were curated, and so that allowed for an opportunity for machine learning to come in and do something important in the field. One critical aspect of all of these is that verification is easy, meaning if you generate a dog image, for instance, the one you can see on the right, you can immediately see that it's not a proper dog. And so humans can easily verify whether the machine learning algorithm is working or not. And that's going to be important. I will talk about this later again. In other cases, when you don't have a lot of data, instead, you have good simulators. So they can generate data on the fly for you, even though the space of possibilities is really large, like chess or Go, you can still play and interact with the simulator and learn a lot about the process. Okay, so now I'm going to talk about machine-guided design, which is an emerging field, and the challenges that you face when you try to go into this field. Some examples of these, not strictly falling within discovery or design, but again, things that you have to do in the physical world, like self-driving cars, have different challenges to the ones that I just described. It's clear where things like chemical synthesis or biological drug discovery, where you're trying to design a protein or an antibiotic, or in healthcare, where you're trying to diagnose a patient or suggest some particular treatment. So one aspect is data is often noisy and heterogeneous. Data between patients can be very noisy, or data that you measure in laboratory can be noisy, or driving on a street can be very chaotic and difficult to box into a particular thing. Whereas you have very clear sentences in internet data, you have very clear headshots or images of that in internet data. Another aspect is data is expensive, and that means your data sets are much smaller, or they take a long time to acquire, which is another way of saying it's expensive, but sometimes you both take a long time to acquire them and you have to spend a lot of money to do it. Importantly, verification is hard, meaning, for instance, if you generate a particular drug candidate, you have to go in the laboratory and actually test it. You can't just look at it and know if it's a correct or functional drug. Sometimes you can eliminate bad solutions, but often you can't know if you have generated a good solution. So that slows down the cycle by a lot. And then the design space can be very complex and very, very large. You don't have good simulators. So unlike chess or Go, which the possible number of solutions or possibilities is really large, more than the atoms in the universe, For protein design, you have the same problem. There are more than possible atoms in the universe, types of proteins that you can design for a protein of size 100. But you end up not having a simulator that can help you interact with that possibility and see what's going to happen. So what is the problem that Dyno is trying to solve? Our headliner is like, we are an AI-powered gene therapy company that makes vectors. So the how is AI-powered, the why is gene therapy, and the what is vectors. And basically, partners come to us and ask for us to give them particular vectors that work for particular gene therapies. Why do we want to do gene therapy? Consider a disease like hemophilia, which affects a lot of people. The reason it happens is that a factor is missing for blood clotting, and that's due to a deficient gene. And so the idea is, if you can deliver the healthy version of the gene into the correct tissue, in this case, say the liver, and make the liver produce the protein that is missing in the body of a patient, then that patient will not suffer as much or not at all from the hemophilia disease. And so there are thousands of diseases like this. The key, though, is to deliver the gene to the right tissue and make that gene produce the product that you care about. Currently, how this is done is with a benign virus known as AAV. It's a virus that cannot reproduce themselves and on its own, and a lot of humans have already exposed to them, and they don't cause any diseases. So we try to do, and the virus is made up of 60 subunits of 735 amino acids that come together and make this ball. The idea is that we can put the gene that we care about into this virus and hope that the gene goes into the particular tissue that we want to treat. However, natural viruses, the ones that exist in nature, they have their own particular preferences for which tissue they want to go to. Say they want to go into the liver, but not the brain. So we cannot use the virus to target the brain for diseases that affect the brain. Another issue is because a lot of the virus has been seen by human bodies and human bodies have generated immunity against them. When you try to inject this virus into a human, the immune system will reject it, basically cancel the virus capsid and clean it from the system, and it won't get to the tissue that you want. So the hypothesis is, if we change the sequence of the virus enough, we can make it go into the particular tissues that we're trying to target. And then we can also make it different enough from the ones that are in nature so that the immune system no longer recognizes it. So you can deliver the treatment, a one-time treatment to a patient, and help them with the disease they're suffering from. Traditionally, the approaches that's been used to address this sort of design is either directed evolution that come from natural models. Basically, you take a lot of viruses, billions of viruses, you experiment them, you inject them into mice, and then you ask which one of them are doing particularly well in going to a tissue. You iterate this experiment multiple times, and then you get the best viruses out of that experiment and use them to try them in clinical trials. Obviously, this requires screening to billions of viruses, and many of them are not going to be successful, at least in the mice. And maybe the thing that you get at the end is going to work in mice, but not in humans. Another approach is rational design, where a bunch of experts sit down together and try to come up with a design that's going to work. This has more successes per design than the first example, but it's still hard to come up with a rationally engineered virus that's going to work all the way. So the number of things you design is often not sufficient for downstream filtering and selection. The idea that we are based on is that if we use machine learning, we can get the best of both worlds. We can design hundreds of thousands of sequences at once with machine learning algorithms, and most of those sequences will do something useful instead of the case of doing a lot of things with natural evolution, where most of the samples you generate are not very functional. And I'll get to that and come back to that in a moment. So what is Dyno's approach? We are a sequence design company. So we generate the sequences ourselves, not the full thing, but basically each step of this goes through our pipeline. We basically design DNA sequences that are going to translate to proteins. And then we produce viruses or vectors based on those sequences, hundreds of thousands of them. And then we put them in mice and non-human primates and collect data from them. So hundreds of thousands of vectors can go into a single mouse or a single monkey. And then we ask which one of them went to the tissue we cared about. That information is collected by the data science team. And then data science team designs a new set of sequences that are going to go through the cycle again. So that's the key aspect of Dyno. We go from experiments to data science and machine learning, and then design the next set of experiments with machine learning. What happens inside the data science, which we are doing sequence design within? First thing, as I said, the data can be very noisy. So we will build a approach to quantify how noisy each data point is and how much we can trust them. And this is a very involved process of being able to exactly figure out how each data point, how much error we expect to see in each data point. Using these data points, we train machine learning models that can tell us the relationship between the sequence and its property or its function. And that allows us to ask the models that we build whether a particular sequence is going to function well or it's not going to function well. Finally, with those models in hand, we can search the sequence space, the space of so many possible sequences, to find sequences that we think are particularly good at a property we care about. So this cycle is what happens inside the data science team. And if you are interested in learning more about it, I have actually, me and a bunch of colleagues have implemented a sandbox, if you will, a programmatic in silico sandbox that you can use to go through these and test your models and algorithms and see if they're doing anything good for sequence design. And it's freely available on GitHub, and you can develop your own algorithms and methods on top of it. So I'm just going to show you a snippet of each of these steps within our pipeline. The first step, as I said, is to measure. And I'm showing you data from publicly published paper in Science a few years ago from our team, and Pierce Ogden, a colleague of mine back then in Harvard, where we measure the effect of changes in every position to every amino acid for 735 positions. And blue here means it did worse than the natural variant. White means it did roughly the same, and red means it did better. As you can see, most changes to the capsid result in a completely broken virus. So if you change the protein, even by a single edit, most of the time you completely break it. So this shows the challenge that we are facing with. We are trying to learn from this data and modify the capsid, the virus, basically, many, many times. And in fact, the exciting story is that we can do this. It just requires very careful modeling. How do we do modeling? I'm just going to give you a very rough overview of the type of models we use and conceptually clarify what they're doing. There are, generally speaking, three types of models we use. The left side, where I call like oracles, they go from sequence or representation of a sequence to a property estimate. You can think about them as a panel of judges which give scores to any sequence that you show them. Second kind is they go from a sequence to a continuous representation or a simplified representation of that sequence. So here I have an image of a cat to a drawing of a cat, which is a representation of the cat, but it's simpler than the original one. This sort of conceptual simplification can help us design better sequences. I'm using cats and dogs here because they're clearer intuitively, but obviously we're doing this with sequences. And the final step is taking those representations and turning them into sequences again. So here is another model example of taking representations of dogs and turning them into dogs. You can think of the same thing. We take representations of sequences and turning them into sequences. So a typical model in our company takes a sequence inside, learns some certain representations or embeddings of them, and also is able to tell you an estimate of whether they're going to do a good job in the heart or in the liver of a particular organism. And then we can have generator models that take these simplified representation and produce new sequences that we think are going to do a great job. The final step is with these models in hand, we are trying to find sequences that do really well. And you can think of this approach is to come up with an algorithm that passes messages back and forth with your models and then ask them how they think about it. If the model is really happy about something, well, we have found a solution. If it's not, then you change it a little bit and see if the model is happier or less happy with it. And you can keep changing it until the model arrives at a solution that it scores really highly. Taking these steps together, we loop back, we send this to the laboratory, and we measure new sequences. We now have more data, we can build better models, we can search the sequence space with more accuracy and better chances of finding the sequences that we want. To summarize all of this, machine learning has been really successful in the past decade. On top of that success, there is an attempt to build design algorithms that make things in the real world. And Dyno is one example of such company that is trying to design things in the real world that's going to be solutions to problems we have. I think this model of combining experiment with machine learning is going to be very popular in the near future. It has its own drawbacks and challenges, and means you have to adapt your machine learning to this condition that your data is generated much, much slower and much more, you have to make better use of the low amount of data that you have. But the approach that we have is trying to have this integrated loop between experiments and machine learning and try to tighten it with better designs and faster experiments. I hope you found this talk interesting, and if it piques your curiosity and you have questions about it, please feel free to reach out to me on LinkedIn. And I'm very grateful that I had the chance to talk to you today.